And Jeremy Rentsch, you can take it away.
All right, thank you, everybody. It's really pretty flattering to be asked to come and give a talk to the ICPS, so I really do appreciate it. I'm going to talk to you a little bit today about the Venus flytrap transcriptome. A little bit of background first. I'm a, lifelong appreciator of carnivorous plants. My love probably started during a field botany course in Ohio when I saw Sarracenia purpurea in situ for the first time with Dr. Barb Andreas, who was the herbarium curator at Kent State University. Your brain just starts asking questions right away. A curious person can't help but ask questions about these really remarkable plants. It hasn't been until much more recently that I became a carnivorous plant biology researcher as well. My work in carnivorous plant biology started with some basic population genetics. That's maybe a story for a different day, but it made me realize that Sarracenia needs, I think, pretty desperately some population genomic approaches to get a really good phylogeny resolved. We should probably talk more about that later at some point. More recently, and what I'm going to talk to you about today, is some work looking at the RNA of the plant, the transcriptome of the plant, to gain insights into carnivorous plant biology. If you don't know what a transcriptome is, it's okay. I have front-loaded the methods a little bit with some information on what that is and why we would care about it. For me, I think the thing that's so fascinating about the botanical carnivores is that they display such an intricate, sophisticated, fascinating solution to a problem that could seem almost kind of pedestrian or banal or almost boring from an ecological standpoint. And that's deficits in soil nutrients for the most part. Today, I'm talking about Venus flytrap specifically, but this applies to most carnivorous plant lineages. The Venus flytrap, for example, lives in waterlogged soil. Waterlogged soil doesn't have a lot of oxygen in it. Microbes that would normally be doing the decomposition and nutrient cycling, well, they're not very active, and so not a lot of nutrients get put into the soil. Likewise, the low pH impacts soil chemistry, and things like nitrogen and phosphorus become much more difficult to take out of the soil. They become bound to the soil particles. The reason I call this a sophisticated solution, especially when we're talking about something like the Venus flytrap, is that the plant itself has to perform an awful lot of functions to ultimately capture and extract chemical nutrients out of its prey. So if we think about the Venus flytrap, they have to be able to detect prey. And in fact, the trigger hairs, as you know, have a mechanism for detecting false positives. The trap itself has to be able to close quite rapidly. Ideally, the trap will reset if it's been falsely triggered. If we've detected prey, we need to hermetically seal the green stomach so we can actually digest what's inside. You then digest the prey, which is a complicated series of enzymes, a cocktail of enzymes by itself. And then, maybe most fascinatingly, the leaf takes on what is typically the function of the roots, the domain of the roots, and that is to absorb the chemical nutrients. And so this is an awful lot of work for simply overcoming a deficit of soil nutrients. That's why I say it's kind of a sophisticated solution to a rather average or almost boring problem, but it's fascinating that these exist. Now, I am very good at killing jokes. It's almost my job, but I think this joke that circulates social media every once in a while is at least fun to talk about because it directly ties into what we were just mentioning. Okay, Venus flytraps do photosynthesize. These jokes all have some element of them that maybe the Venus flytrap is just eating like we do, and that's all they really need to do. Well, Venus flytraps do photosynthesize. They're capturing and digesting arthropod, primarily prey, for the chemical nutrients, the nitrogen and the phosphorus. But I bring this up for more than one reason. The other reason I bring it up is that there's some really fascinating work relatively recently that shows, in fact, the Venus flytrap can use prey-derived amino acids to fuel cellular respiration. Typically, when we think of photosynthesis for plants, we're thinking about fixing sugars. Those sugars are used to fuel cell respiration. But in fact, this research has shown that Venus flytraps can use prey-derived macromolecules for energy, and that is interesting. They certainly wouldn't use it for all of their cell respiration, but I guess it's kind of a waste not, want not philosophy. So what research am I going to talk to you about today? What I wanted to see was this. Could we space out some time points where we sample the transcriptome of the Venus flytrap? Again, I'll tell you what that means, in a way that gives us some insight into what I hypothesized would be a nice, elegant time series that represented prey capture, prey digestion, nutrient absorption, and maybe the prey digestion would even be in a series where chitinases are released first and then the proteases and lipases. That was the hypothesis, and so we set up some time points for RNA isolation that would allow us to answer that question and generate some new insights into the biology of these plants. So our sampling design looked something like this. What we would do is feed a Venus flytrap mealworm effectively, and then wait for some specified amount of time from 5 minutes up through 72 hours. When the specified amount of time had passed, we would harvest the trap, isolate the RNA, sequence the RNA, and then do comparisons between and I guess among time points. I'll talk a little bit more about the methodology, but this is the basic experimental design. Each of these time points were repeated 4x so that we could be confident in the data. And we didn't stop there. We also mirrored a few of these time points with traps that had been triggered but without prey to see how the transcriptomes of a trap with prey varied from the transcriptome of a trap that had been closed for an equivalent amount of time but didn't have a prey item inside. A quick, I guess, tutorial or reminder about what the central dogma is and why we might want to sample RNA to begin with. So transcriptomics can be thought of as the study of all of the RNA in a cell or tissue or organ or organism at a certain time and under a certain condition. This is to be contrasted with something like a genome. That's the DNA or the proteome. It's the complement of all of the proteins in a cell or tissue or organism. If you think about the central dogma states that information flows from DNA to RNA to proteins, with RNA serving as kind of the intermediate. It's what we get between DNA and proteins. And there are reasons to study RNA versus DNA or proteins. There's reasons, in fact, to study all three of these macromolecules. And so as a quick reminder, DNA you probably hear about a lot. DNA is kind of the king of macromolecules when it comes to estimating phylogenetic relationships, constructing genetic maps and karyotypes. For conservation genetics, you want to look at DNA. DNA has neutral markers that evolve more rapidly than markers that might be under selection. And this gives you a chance to look at neutral variation, which is a good indication of divergence between or among populations. So there's plenty of reasons to study DNA. Likewise, studying proteins, all of the proteins together we call the proteome. You study proteins because that's the actual expression of the gene. Most genes express themselves as a protein. There are regulatory sequences as well. But most coding genes will code for a protein, and that protein might be structural. It might be a surface marker on a cell. It could be an enzyme. And so there are plenty of reasons to study proteins. They can be difficult to work with. It can be kind of a low throughput project or experiment, but plenty of reasons to study proteins as well. So you might wonder then, why would you study RNA? Well, RNA is also gene expression. Upregulation of RNA will eventually likely yield those proteins. But what I find most interesting about RNA is the speed with which the transcriptome changes. It can take hours, conservatively, a couple of hours to, let's say, treat a plant, a heat stress treatment or something like that. It can be a couple of hours until you see actual heat stress proteins in a cell. But it's a matter of a minute or less to see changes in the transcriptome. So the way I think about it, and I've never been good at analogies, but looking at the transcriptome is almost like looking into the mind of the organism. You see what it wants to do or what it's going to do. There are some exceptions to this. There are post-transcriptional or pre-translational modifications that can occur such that the RNA never gets translated. But for the most part, RNA reacts very quickly. The transcriptome reacts very quickly and is a really fascinating window into what the plant is going to do, or very likely going to do. Now, here's where RNA is so much different than DNA, is that in our bodies, for example, except for the rogue mutation here and there, all of the DNA in all of our cells should be the same. Again, save the rogue mutation. The transcriptome, or the RNA, isn't like that at all. The RNA in your epithelial tissue versus your nervous system versus your liver versus your kidney will be different all the time because they have to do different things. They have the same DNA blueprint, but they're using them differently. Thus, the transcriptome has to be different. Likewise, the transcriptome changes with age, life history characteristics, whether you're in a reproductive mode or not, whether you're stressed or not, and what kind of stress. So the transcriptome tends to be dynamic and very quick to respond to different environmental conditions. So what did we test? What RNA did we test? There's a few things you could do here. We sampled full traps. So I would feed a trap, wait a certain amount of time, excise the entire trap, cut out the prey item, and then throw the little bugger in some liquid nitrogen to cool it down real quick. That's one way of approaching it. The other way of approaching it would be an even more targeted method where perhaps what you're really interested in are what the glands of the Venus flytrap are doing. That's probably where a lot of the magic is occurring, of course. So another option would be to excise some of those glands, do RNA isolations from just the glands. You potentially lose some data. When I look at the whole trap, I get to see how the mesophyll is responding potentially, for example, although that has to be teased out. But testing the glands only, or isolating the glands only, would certainly give you more confident data about what just that structure is doing. So this is important, and there's no right or wrong answer to it necessarily, but it does influence how downstream analysis has to occur. I thought it'd be interesting just to, for 2 minutes, talk about what this lab work can look like, because it's very easy to say we sampled the transcriptome or isolated RNA, but what that actually means, well, it's probably a little different for everybody, what you're imagining this meaning. So what we do is, like I said, we feed a trap. We let the trap stay closed for however amount of time, the 5 minutes through 72 hours, cut off the trap, excise the arthropod, and then throw the trap into liquid nitrogen where it can be kind of ground down into a really fine powder. That looks really haphazard, but it's a few things are important here. One is that RNA is very delicate. I tell my students that if you have tissue kind of dying on a countertop, the DNA will be the last thing to degrade, kind of, and the RNA will be more or less the first thing to degrade. There are just ubiquitous RNases everywhere, and it's prone to some self-destructive behavior, in fact. The important thing here is to keep the tissue cold. So you throw a trap into liquid nitrogen, get real cold, powder it, and then before it comes back up to room temperature, you can throw it in some lysis buffer, heat it up, and this liberates the nucleic acid. So that first step, the homogenization process, is really just to open up the surface area of the trap we're looking at. The cell lysis stage is for ripping apart the nuclear envelope, ripping apart the plasma membrane, and making sure that all your macromolecules are able to float around your test tube, your microcentrifuge tube. So on the right-hand side here, we can see a warmed up mix of buffer and powdered venous flytrap that has proteins and lipids and sugars and nucleic acids floating in it. Probably the most interesting part of this is the phase separation chemistry. So what you can do is the cell lysis buffer is an aqueous buffer. And so if you hit that buffer with a strong nonpolar solvent, some mix of like chloroform and phenol, what you can do is gently homogenize your microcentrifuge tube. You're giving all of your macromolecules a chance to decide where they want to go, depending on where their favorability is. Fats, for example, are going to stay dissolved or going to dissolve in the nonpolar solvent because they are nonpolar. You centrifuge the heck out of this thing, and you end up with a couple of layers or a couple of phases. The topmost phase and the clearest phase is called the aqueous phase. That's the remainder of our buffer. And luckily, nucleic acids are water soluble. So at this stage, you can just take off this aqueous phase with a micropipetter, and you've separated out already most of the fats and proteins and sugars. So this does most of the nucleic acid purification for us. You then, and I actually used a slightly different method for the venous flytraps, but I wanted to show you this picture. The little snot-looking guy inside that microcentrifuge tube is in fact concentrated nucleic acid. So that's what nucleic acid looks like if you have enough of it. That could be then treated with DNases if we're doing RNA work to degrade the DNA, so the DNA doesn't get greedy and take up space when we're trying to do sequencing. And then it can be just suspended in water or some kind of buffer for downstream analysis. It's probably a little much to talk about the sequencing of RNA, but hopefully that's at least interesting to see what some of the lab work looks like. So I wanted to spend a little bit of time just generally talking about what some of the results were here. So I'll remind you that we have these time points that hopefully represent a nice time series of prey capture, digestion, and perhaps absorption. And we also have some matched time points that are traps that have been triggered but without prey. Those were at 5 minutes, 1 hour, and 24 hours. So I wanted to share, before I get into the data, I wanted to share just a general discovery, and one that I kind of think is maybe the most interesting of the whole study. And it comes from this matched 5-minute time point, 5 minutes with prey, 5 minutes without prey. And really interestingly, we see that the transcriptomes, and this is only the case here, the transcriptomes at the 5-minute time point are identical, whether or not the plant has prey or no prey inside of the trap. Statistically identical, no difference at the 5-minute time point in gene regulation. But really interestingly, and we'll see this when we look at some of the data, the trap does appear to be primed for digestion. So at the 5-minute time point, we already see signals that the cysteine endopeptidases and chitinases, for example, are being upregulated. Now, not to give this work too much credit, but I think maybe we can say that this officially makes the venous flytrap an optimist, because it seems to not know whether it has prey or not. Otherwise, the 5-minute time points would look different between those two treatments. But certainly, the venous flytrap is optimistic that it has caught something, and I kind of think that's nice. At the 1 hour time point, there were actually relatively few differences. This is very speculative. Okay, the 1 hour time point in traps captured with prey is kind of going off like gangbusters. Without prey, there aren't too many genes being differentially expressed, but many of them that are being differentially expressed are genes that seem to be targeted to the chloroplasts. Very speculative and just a hypothesis, but I actually think then by the 1 hour time point, those traps that have been triggered without prey maybe are already starting to repair some of the oxidative damage that occurs during trap closure. Trap closure is pretty darn damaging to a Venus flytrap. It generates a ton of reactive oxygen species. Reactive oxygen rips apart chlorophyll like it's its job. And so for the trap to reopen and be healthy enough to close again, likely there has to be some repair done. That's very speculative, but I think it's what's going on in the 1 hour time point in the traps triggered, but without prey. I want to acquaint you with the data, what the actual analysis looks like for this study. But another conclusion here, and something that I find to be fascinating, is that throughout the course of our time series of digestion, the 5 minutes to 72 hours, we see that about 50% of the Venus flytrap coding portion of the genome is upregulated following prey capture. In other words, about 10,000 genes over just over 21,000 genes are found to be differentially expressed at some point during this experiment. That means that, of course, the Venus flytrap is putting an awful lot of resources into this. The genome is well honed towards this process of prey capture and digestion. I'm going to share a few more findings, but those findings will require being just a little bit oriented with these data. In this data, we can see kind of three main figures, A, B, and C. Figure A, we're dealing with 10,000 genes, a little north of 10,000 genes. So we can't talk about just each gene. Instead, the genes get kind of collapsed into modules that represent similar patterns of expression. So, for example, what's been named module 3 is the most gene-rich module. These are all genes that share a similar expression profile across the experiment. There's about 3,000 differentially expressed genes in this module. And if you see the box on kind of the bottom right, that is more or less the expression profile of module 3 with, you can see, kind of, since we're collapsing so many genes into one graph, it's got kind of a lot of noise around the signal. But what we see almost definitively, I guess, with module 3, and another thing to note, is that module 3, the most gene-rich module, in fact, either has peaked expression at 72 hours, or in fact hasn't even peaked expression at 72 hours. That already suggests a transcriptome that's so rich and so dynamic that it goes beyond, perhaps well beyond the bounds of what I was able to test here. What you're looking at, you can just think of as average gene expression. So what this is you take all of the transcripts that are expressed throughout the course of your experiment, you get an average for what they look like across time points, and then you can see how that average varies, either higher or lower, throughout the time series. And so we can see that module 3 is a module composed of genes mostly that are just spiking or haven't yet hit their peak expression value at the 72-hour time point. But mostly just think of that as just gene expression, just statistical gene expression. Now, if we were to look at the gene classes that are in module 3, you can see where it becomes difficult to tell too broad of stories. I'm going to focus on kind of smaller stories because module 3 has many gene classes represented in it, and it's hard to narrow it down to maybe a single function. So what I would like to do is kind of talk about some individual genes that we see in different classes and kind of talk about how those make sense in the story of the Venus flytrap. So looking at module 3, for example, I see the chitinases, which are something that I mentioned or talked about just a little bit earlier. If you're familiar with chitinases, these of course are enzymes that break down chitin. Chitin makes up the vast majority of the exoskeleton for arthropods, and so it makes an awful lot of sense that the Venus flytrap would see upregulation of chitinases during prey capture and digestion. What's interesting is the majority of chitinases seem to be expressed in module 3. Recall that module 3 doesn't see peak expression until 72 hours in and starts at pretty low expression. So that is almost contrary to what we would expect chitinases to be doing in the Venus flytrap. But if we look at the chitinases, and there's only one each in modules 10 and modules 9, they fit an expression profile that we would associate with carnivory, or that we would associate with the degradation of arthropod exoskeletons. In other words, modules 9 and 10 start off with a quite high expression value, higher than average, and then drop over the course of 72 hours, more or less. Those are consistent with carnivory. You dump in some chitinases, you let them do their job, you dump them in early, and the exoskeleton gets at least partially degraded. So then, what's going on with the module 3 chitinases, where the chitinases are in fact more robust? Well, I think we know that there are dual roles for chitinases. Carnivorous plants share chitinase activity. They don't get that from nowhere. Plants are familiar with chitinases and have chitinases even in non-carnivorous plants because they're constantly having to fight fungal pathogens. Fungi have chitin in their cell wall, the same way that arthropods have chitin in their exoskeletons. They're chemically just a little bit different. They have certain modifications, and so the same enzyme wouldn't have peak effectiveness or efficiency with both kinds of chitinases. So likely these have been kind of sub-functionalized in some way. The chitinases in module 3, therefore, are very likely chitinases that are meant to keep the trap from putrefying, which is something the Venus flytrap has to worry about. You capture some prey and you digest it. It's rotting inside your trap. The trap is maybe taking a little tissue damage too, because the process is quite destructive. The last thing you want is to get a bacterial or fungal infection that essentially comes in and steals your meal. So chitinases get upregulated, I think, as a matter of course, to defend against fungal pathogens that might come and take our hard-earned prey. Likewise, something interesting is that most of these modules and all of the gene-richest modules are also packed full of lipases. Well, most arthropods aren't that fatty. They're very proteiny and very chitiny, but they're not that fatty. Of course, we probably know the answer to this too, and that's that the Venus flytrap glands run off of lipids. That's rare for a plant. Plants, of course, prefer starch and sugars. Well, starch for their long-term storage molecule, and sugars to burn for ATP. But there's the occasional fatty plant tissue. You think of an avocado, or you think about seeds that are packed full of fat. The caveat for plants is that they don't really send fat around their vascular tissue. So if you're synthesizing fat somewhere, it's more or less got to stay there. Fat's very high in calories, and so packing these glands full of lipids gives them kind of more calories per gram for this energetically expensive process. So, well, we see an awful lot of lipases throughout these most gene-rich modules, and probably not all of them are to fuel the glands. It's a little hard right now to tease apart which lipases are for which exact activity. So let's think about one of the proteins or sets of proteins most closely associated with carnivory, and that's the cysteine endopeptidases. These are in fact named Dionaea. They were, at least a few of them were discovered in the Venus flytrap. Now those appear in module 9 and only module 9. So if we take a look at module 9, it might give us some insight into how the Venus flytrap is thinking. And this I found at least a little encouraging because if you recall my initial hypothesis, it's that we will maybe be able to uncover a really elegant pattern of prey capture, digestion, and absorption. And the regulation of the cysteine endopeptidases seems to give some credence to that, with those being in their highest expression at the 5-minute mark. Again, Venus flytrap is an optimist, certainly hopes it caught something. It can take a few hours to go from the transcriptome to fully realized proteins. So this is a process to get started a little bit early. So this may lend some credence to that elegant time series of digestion. But of course, there's still a lot of data, I think, to tease apart with 10,000, over 10,000 differentially expressed genes. You could probably find justification for any hypothesis you wanted to justify. So while I'm looking at the data, I feel like I see some trends emerging, and I do feel like there is some pattern to the series of capture and digestion, but certainly more work has to be done before we can call it elegant. We've seen another kind of interesting thing is we've seen the upregulation of certain nutrient transporters at the 1-hour time point. So some of the big transporters for nitrogen, moving nitrogen into the trap, effectively acting like the roots. These are genes that are constitutively expressed in most plant roots. They're just expressed all the time, but they don't get expressed in the Venus flytrap until it's got something to absorb or thinks it's going to have something to absorb. But this does kind of lead me to another question, and it's this: what does the Venus flytrap look for to ultimately decide if it has suitable prey? I thought I knew the answer to this. I'm not sure I know the answer to this now. And I say this because if suitable prey is decided by the continued generation of action potentials in a closed trap, you would probably think that the plant, the trap, knows by the 5-minute time point that it does in fact have something struggling inside. In other words, if that were the case, the 5-minute no prey time point should be statistically different than the with prey time point. So maybe it's not just action potentials. There's some, I think, traditional wisdom that perhaps it's the liberation and detection of prey-derived macromolecules that kind of keep the digestive process going, or helps the Venus flytrap decide whether or not it has prey. But it also seems very fast that at the 1-hour time point, there would be prey-derived macromolecules. That seems very fast to go from transcriptome to realized proteins to beginning the digestive process. So there's a little more to untease or untangle or disentangle here too, I believe. I think in terms of the next steps for this work, what I would like to do, it's clear to me with all of these genes and all of these time points that there's a lot of data to still be discussed and described in this data set. And I think the easiest way to do that would be, okay, adding data to it, frankly. What I'd like to do is look at Drosera and do kind of a scaled down experiment in the sundews, looking at pre-feeding, 5-minute and 1-hour post-feeding. And I think that that'll let me develop something like a Venn diagram of gene expression, where maybe because the Venus flytrap and the sundew share a common origin of carnivory, perhaps it's going to be possible to separate out some of the genes involved in the active portion of Venus flytrap capture. because likely Drosera won't display those same genes. And so by looking at what these species share and what's different, I think we'll be able to pull out even more insights from the existing data set. I then plan to work out in kind of concentric circles to describe how independent truly is when we're talking about botanical carnivory. But that's going to be, I think, a very long-term play. I tried to go light on the data. I didn't want to talk too much about that, but suffice to say, I'd like that to be very transparent. And so all of these data are available online. The raw sequence files are available online, or the short reads rather, available online. There's also a GitHub that my colleague created that has all the scripts used to do data analysis. So these data are all out there for anybody who's curious in looking at them. I do have a lot of people, of course, to thank for this. I have some wonderful colleagues who helped me with the work, Jim Liebensmack and Summer Rose Blanco, both of University of Georgia, ICPS's very own Carson Trexler for some talk feedback, Kenny for the invitation today, a whole host of people who helped with genomics and data curation in one form or another. My home university, Francis Marion University, I had the funding to do this through my sabbatical project. And so this work was funded by my home institution. Thanks, of course, to everybody who listened. I hope you learned at least one new thing about the Venus flytrap today, and I really do appreciate your time. I understand Kenny has some business to discuss first, and then I'll be happy to take questions after that. Thank you.
All right, thank you so much, Jeremy. And you do have a couple of questions in the chat box if you want to peruse them now as I talk briefly about the ICPS, and then I will read them for everybody who is watching this as a recording. All right, thank you, Jeremy. Great presentation. And you presented, of course, for the ICPS, the International Carnivorous Plant Society. We have 1,404 paid members currently. And we are a group of scientists, horticulturists, conservationists, backyard growers. So we span the world and we span lots of different topics and interests regarding carnivorous plants. As of a couple of years ago, we like to host monthly webinars. And in the next couple of months, we have 2 webinars a month, and they are available for free to the general public. And we would love for you to RSVP to those. And if you can't attend them live, you can watch the recordings on YouTube. They are held at different times of the day and different days of the week because we are international and we want to get as many people involved as possible. If you are interested in presenting, you can contact me. My email is down there at the bottom. And we're interested in people who have big collections, small collections, who are doing research on carnivorous plants, people who have done field trips for carnivorous plants. Jeremy mentioned that I think one of his first carnivorous plants he saw was Sarracenia purpurea in the wild. And I was just in Halifax, Nova Scotia last week in September, and I saw fields of blooming Sarracenia purpurea, and it was quite remarkable. If you go to our website or if you go to our YouTube page, you will see that we have 5 animated videos about carnivorous plants. They are titled How to Grow Carnivorous Plants Indoors, How to Grow Them Outdoors. One of them is on how to acclimate new plants. One of them is what is a carnivorous plant. So you can see that they're animated. They're great for children, but they're also great for people just getting into the wonderful field of carnivorous plant botany. On the first Wednesday of May every year, it is World Carnivorous Plant Day. So for the past 4 years, we've had World Carnivorous Plant Day, where we post over 20 or 24 videos every year on that day to celebrate carnivorous plants. We've had celebrities participate. We've had growers. We've had people from lots of different countries, Brazil, Australia, Japan, lots of European countries, and then lots of people in the US, Canada. And we would love for you to be involved in the 2025 World Carnivorous Plant Day. You can also message me if you're interested in presenting. And if you go to our YouTube page, you can watch hours and hours and hours of all the past presentations. Some of them are, I think the shortest ones are 5 to 10 minutes, but some of them go upwards to an hour. So if you ever need any information, it's probably there. Earlier, I mentioned that we are a paid international organization. If you can help support us, that would be great. The more members we have, the more authority we have. So if there's a conservation initiative that you want to take part of, your membership dollars can help us fund those things. And I'm going to talk a little bit about that in a moment. But also, one of the benefits of the ICPS is if you become a member, we have a seed bank, and it's as good as our members make it. So when you have surplus seeds, you can donate your seeds, and we can ship to many parts of the world. You can contact our seed bank manager, Mark, and he will tell you if he can ship seeds to your country. And the real highlight of being an ICPS member is the Carnivorous Plant newsletter. That's why our society started. All the way back in 1972, we've had a newsletter. Of course, now it is full color, glossy, beautiful magazine. It is filled with scientific reports and findings. It's filled with field trips. This is the official place to register a cultivar, a cultivated variety. So if you think you have something really fancy and you want to name it, you have to go through the ICPS. And you get this beautiful quarterly newsletter, full color. And when you become a member, the best part is you get access to the last 50 years of newsletters. So you can see the actual original writing of a specific cultivar, or you can see when this plant was discovered in the wild. And also, we're always looking for contributors and writers. You could be somebody who repurposed a flower display case into a grow chamber, or you could be a Ph.D. at Harvard. All right. Every August, we have the International Carnivorous Plant Society's Carnivores in the Classroom. This is where we fund up to 50 teachers around the world with $150 worth of carnivorous plants for their classroom. And one stipulation is that the plants have to be inside the classroom. So we're not really trying to fund people who want to have like a bog outside of their classroom or on school property. We want the plants to be actually in the classroom where kids can interact with them. And I think Jeremy was mentioning, like, I think maybe in college, he got that lure of, he got entrapped by the carnivorous plants. But we want to do that for all ages. So these funds are for K through 12 kids, because we want them to be excited by carnivorous plants, but we also want them to be excited by conservation and botany in general. In order for teachers to get those grants, they have to create a tiny lesson. And this is just a sample. We have not updated the 40 lessons from this year, but we have lessons for K through 5. We also have lessons for middle school, and then we also have lessons for high school. And almost all of them are a PowerPoint with the PDF, or a PowerPoint and a Word document, a PowerPoint and a Google Sheet. So you can edit everything because you know your students the best, and then you can utilize it in your classroom. If you want to support our education program, you can go to carnivorousplants.org/donate. These are the nurseries that have funded all of the teachers for the year, for this school year. So last month all the way to next year. And then in the middle, we have Carnivorous, who created, with the help of ICPS, 5 3D models of carnivorous plants, and each teacher will receive one of these 3D models. And you can purchase them from Carnivorous, or very soon you can go to our website, carnivorousplants.org, and you can get the free instructions. So if you know somebody who has a 3D printer, you can print them whatever color and size you want. And if you want to help support our conservation or our education initiatives, we have an international store. This is drop shipping, so they will print it locally and then ship to you. And all proceeds, of course, benefit our non-profit. And I just wanted to put this little sample of articles that are in the Carnivorous Plant newsletter. And I wanted to highlight this one, and I didn't know Jeremy was going to mention it, but Carson Truxler wrote last year about expanding the ICPS's scope about how we can impact endangered plant species. So if you want to help save carnivorous plants, the very plants that you're growing in your bedroom, you can contact Carson. He's the conservation director, and we can come up with projects for 2025. Every other year, we have a conference. In this past May, it was in Vienna, Austria. We had the most people ever attend. We had over 180 people attend, and we do the conferences every other year. The next conference is going to be in 2026, and it is going to be in the Americas. So it could be in North America, but it could very well be in Central or South America this year. And we rotate. So last time was Europe, now it's the Americas, then next it would be Asia, Australia, and then go around the globe. And thank you for that commercial. Okay, Jeremy, we got from Raphael. Would you expect the same results as the plant transitions from spring to fall? This is in regards to starting off with more stored energy versus accumulated resources.
Yeah, it's a really fun question. I think because of what I sampled, I would probably expect results to be kind of similar, pretty similar, unless there's the fact that perhaps the glands get depleted of lipids and maybe have to attempt to move to a different energy source. But I think there's plenty to study in terms of their dormancy or not. But I think results would be similar regardless of time of year here. It's a fun question.
Alberto asks, are there any pigments genes related, triggered by the food?
I had to put on some blinders with this because there were so many genes. A good example, I don't know the answer to that. A good example of this is hormone signaling is really important in this, especially with the hermetic sealing of the trap. But those networks are so complicated that at this point, I didn't feel like I had the ability to kind of disentangle what the story with the hormones were, for example. So I kept the analysis here pretty tight to some of these proteins we associate with carnivory. So I don't know the answer to that, other than I didn't see those, but they might be in that data set somewhere.
All right, Fernando, who has been with the ICPS for a long time and helps edit the Carnivorous Plant newsletter. And he presented at the Vienna conference. And one day he will present a webinar. He says, apologies if you mentioned this, but I missed the beginning. It appears you sequenced mRNA, not whole transcriptome, correct? How many million reads per sample did you sequence, and with which sequencing technology? Because I know Fernando, he went to Japan to learn all of this.
Oh, very good. Okay. Yeah, we used an Illumina NovaSeq. That's the machine, at least it was run on. I'll be honest, the transcripts per million, the actual data on the number of reads we got, it's such a staggeringly ridiculous number that no person would know what it means. It's like more than the number of stars and anyway, I don't know that number offhand. It's definitely in the supplementary data. And I wish I had maybe looked over that before the talk, but we used an Illumina NovaSeq for the actual sequencing. Got, of course, a bunch of short reads that then ended up being mapped back to the draft Venus Flytrap genome. So we could calculate transcripts per million, but I don't remember the actual number of reads that was produced here, other than it was some staggeringly ridiculous number.
And because this is shop talk, Jeremy can privately message Fernando.
Oh, sure. Yeah. Okay. Yeah. I'll send the supplementals along for sure.
And Jeremy, before we began this webinar, Maxim, I was talking about you. Maxim is from Slovakia, and he did the presentation a couple of months ago. And he says, splendid talk. Do you expect similar timing of capture, digestion, absorption phase and sundews versus the fly chip?
Yeah, that's a good question too. I don't think I can expect anything. I honestly thought with the way I spaced those time points, I would see a much clearer signal than I did. There's some fun stories to pull out, but I feel like I'm still working on this data set. So it would be arrogant of me to say I expect a similar pattern. But my hope is, since they share an origin of carnivory, although slightly different modes of carnivory, hopefully they will be close enough that I will be able to find some comparisons to anchor into. That's only my hope. I can't exactly say I expect it. I'm just keeping my fingers crossed.
And I think Maxim would be a great contact for you because he's on the other end with sundews doing similar work.
Absolutely. Yeah, I need all the help I can get.
All right. Well, thank you everyone for a wonderful attendance rate. And thank you, Jeremy, for a great presentation. You have several thank yous and nice classes in the comments. And we can have one last question. How does UV light affect the trap's digestion?
Oh my gosh. I didn't do that test, so I would have to, I don't know. I would have to actually do that experiment, I think. Yeah, unfortunately, boy, I really am batting zero with these questions, I feel like.
Well, I think that's the point of science. You share your results and then people say, well, what about this angle?
Good point. Yeah, that's okay. It just stimulates more questions, which frankly, this whole project did for me is generate more questions and answers, but some answers too.
Thank you for that. And thank you for the presentation. And we look forward to everyone attending our next webinar. Thank you, Jeremy.
Thank you.
It's not a surprise that gardeners, educators, and scientists are fascinated by these unique plants. The International Carnivorous Plant Society, or ICPS, not only love these plants, but welcomes growers just getting started all the way through professional scientists. The ICPS even started an annual World Carnivorous Plant Day to celebrate them. The free online event is held the first Wednesday of May. Take a look around our website and you'll find historic documents about carnivorous plants, growing guides, free educational resources, and more. Have questions? Ask. We don't bite, but our plants do.